Today's science and technology news speedrun is very much a science thing. Why is that still flickering? Today's science news speedrun, that flickering's even worse. I do need to replace my USB-C cable that goes from my display card to this little monitor. It clearly can't handle the HD resolution being pumped through it. It's just struggling to move with the times, evidently. I'll replace it. But in the meantime, Science Daily tells us yesterday, scientists discover a single cell builds a brain with 170 billion cells. And the reason why I said that in a bit of a sarcastic tone, just a bit, was that this is overstating it. It's simply a theory. They haven't discovered this. But then, I suppose if the theory turns out to be true, then discovery ahoy. So how does a single cell build a brain with billions of precisely organized neurons? Researchers suggest that brain cells use their lineage, their cellular family tree, as a kind of positional map. Yeah, I could see that. It's no coincidence, I'd say, that they've included a stock image of brain cells and neurons looking rather like a tree, because they kind of are. Cells that come from the same ancestor stay near one another, helping the brain to organize itself without relying solely on chemical signals. And it's a surprisingly simple rule. Brain cells sticking close to their ancestors may help explain how a single cell builds an intelligent brain, or a brain, depending. A human brain starts as a single cell, which itself is pretty amazing, let's face it. Over time, that lone cell gives rise to an extraordinarily complex organ containing roughly 170 billion cells. One of the biggest questions in developmental neuroscience is how all of those cells end up in the right places to form a functioning brain, almost as if there's some sort of massive blueprint ahead of time. And yet, where's the blueprint? Who's directing the individual cells? Each one to be this thing, the other one to be that thing. That's DNA. Probably got something to do with it. But still, researchers at Cold Spring Harbor Laboratory now believe the answer may be surprisingly simple. I think they said that. Have they said that? So they said that there. It's surprisingly simple, and as it turns out, it is surprisingly simple. It's confirmed. Their new work offers insights into how the brain organizes itself during development and could eventually influence research in fields ranging from biology to AI. It's a bit of a leap, to be honest, but I could see that eventually. So how brain cells determine their identity. This is the thing. How does, out of 170 billion cells, how does each one know that it's the one that's meant to be like over there and not the one that's over there? Stan Kirstjens, apologies for the pronunciation there, a postdoctoral researcher in Professor Anthony Zador's laboratory, explains the challenge in terms of positional information. So the only thing that a cell sees is itself and its neighbours, he explains. But its fate depends on where it sits. A cell in the wrong place becomes the wrong thing. Yeah, because its identity isn't just what it is, but also where it is, what else it's connected to. And the brain doesn't develop right. So every cell must solve two questions. Where am I? And who do I need to become? That's pretty profound. It's true of life for the rest of the human as well, not just the individual brain cells. So in a study published in Neuron, good choice, Kirstjens, Zador and collaborators from Harvard University and ETH Zurich propose a new theory describing how the developing brain achieves blah, 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 blah. Chemical signals. For decades, scientists have largely believed that cells communicate positional information through chemical signals. According to Kirstjens, that explanation works well, but only in relatively small systems with limited numbers of cells. But the developing brain contains billions of neurons that each has to arrive at the correct location because chemical signals weaken as they travel. Researchers have long wondered how cells located deep within a growing brain can accurately determine where they are. There's a decay function there, isn't there? Clearly something else is going on, and it might be something else plus chemical signals. They work together. It doesn't have to be one or the other. Kirstjens suggests that part of the answer may come from a process that resembles the way that human populations spread over generations. This is very similar to human demographics. Descendants in a particular area tend to settle near their ancestors, descendants in one place. So if you have a village in Anglo-Saxon England or something, people just tend to stay within that area, or quite often just in that village. They live their entire lives there. They have children there. The children grow up and also live their lives in that village. And it's pretty rare for people to actually move outside that area. Even nowadays, you get the occasional adventurous one who goes off and explores some other part of the world. But generally, people tend to stay put over multiple generations. And so the theory here is that the brain cells are doing essentially the same thing. The team have essentially built a lineage-based model of scalable positional information. Is that in here? Yes. So a lineage-based model of scalable positional information. To investigate the idea, they first used theoretical calculations to explore whether the concept could work. Next, they examined patterns of gene expression in developing mouse brains, looking at both individual cells and large cellular groups. Finally, They tested the model in zebrafish. I was just checking because I wondered if that was the name of some analytical software of some sort, but it is literally zebrafish. And they found similar results to the mouse brains, suggesting that the mechanism may operate across brains of different sizes. Yeah, if different brains that are essentially brains just developed and grew using an entirely different mechanism, that would change a lot of things, I think. The findings indicate that chemical signaling and cellular lineage may work together to provide positional information during development. So what are the implications? Maybe it isn't enough to just know this, assuming it's true. How can we put this to use? Although the research focuses on the brain, Kirstjens says the underlying principle could apply to many other developing tissues, including tumours. The theory may also have relevance for future self-replicating AI systems. Great. Von Neumann machines, named, by the way, after John von Neumann, who was actually studying the idea of self-replicating machines back in the 1940s. Finally, his day has come. Soon. Just as brain cells can inherit information across generations of cells, future AI models that pass information from one generation to the next could potentially rely on similar organizational principles. Perhaps the most significant implication is what the work could reveal about the intelligence itself. Understanding how a single cell develops into a highly organized brain may help scientists answer some of the deepest questions about the mind. The brain somehow makes us intelligent, Kirsten says. How did it manage to accumulate this capability? Not just over its developmental time, but over evolutionary time. This is one piece in that big puzzle, he says. Could there be a question there of evolution, as in random mutations, random events occurring and survival of the fittest and so on, versus intelligent input of some sort? Ooh, lots of other interesting articles here. I can recommend sciencedaily.com. It's pretty great. And that's basically it. Just a quick speed run today through a particular science news item. I do hope you're liking the new format, by the way, and I'm trying not to lean forward too much and have a looming forehead in the camera view. I keep trying to remember to lean back instead of forward. And so thanks so much for watching this far. Please do leave a comment, let me know what you think, and do watch the next video in the Daily Science News Speedrun playlist. And in the meantime, do check out mafftopia.com. It's a portal to YouTube, which is probably where you're watching this right now, and my page on X. We're also over on Rumble, although to be honest I haven't been putting up videos there. I really ought to be. If you like Rumble and you're signed up, do go and check that out. If you follow on there, that'll be a signal to me that I really should be putting up videos there too. Also on this page though, we do put in artifacts and things from things that we've been working on within these videos. So for example, the Angine de Poitrine music science deep dive that I did with Save with Crypto, that involved an AI vibe-coded wazometer, the rhythmic pulse visualizer, which we use to break down the individual beats per bar, and obviously of course the microtones as part of that video. You can actually try out the rhythmic pulse visualizer yourself over at mafftopia.com. Other things, if you're into trading, as in like crypto, forex, stocks, that kind of thing, check out this page, which has a price patterns widget, basically for learning, essentially that. There's going to be a lot more stuff added to this as well, and then a bunch of text you don't really need to read. I wouldn't recommend reading that, it's all a bit rubbish. Hopefully we'll get us showing up in some Google search results, that sort of thing. And that's about it. Thanks so much for watching. Do like and subscribe, all the good things, and see you next time. Bye! 電子の海へと取り込め進化の先へ未来はすぐそこにマフトピアマフトピアクリプトで世界を変え